So first, in case anybody hasn't seen this yet, especially people new to Kinarms, Dexterit-E Explorer is free software. It's available to you from our website. It runs on Windows and Mac. You can run it on as many computers as you want. You can run it on the Dexterit computer to look at data you've just collected, or if you want to put it on your laptop and take data, take your data from the Dexterit computer and import it over onto your laptop, that's totally possible for you to do as well. Within Dexterit, there are some export functions that will export basically a readable database, and then you would just need to point Dexterit-E Explorer at that database, or use under File, there's import subjects, import exams. Either one, those that'll work for you. But just be aware, this is free, so definitely go and download it, run it on your Mac, run it on your PC, and don't be afraid to give us any feedback for new ideas. So first off, this is going to look a little bit at least like the subject database that you see in Dexterit. So here you can see all of your subjects, and you'll be able to see all of their exams. I'm going to find myself. Oops. McLean. There we go. So this is a couple of sets of exams that have been run on me. I'm going to pick one here. So the initial kind of idea behind this tool was to be able to, for you to be able to look at your data without needing to use MATLAB. So there's some nice things you can do here. I'll kind of walk through a bunch of the functions that are available to you. So here we've got you select your exam, and then you can see once I've selected my exam, we've got some stuff here ready to animate, ready to look at. So the first thing, here's your events. These are all of these timed events we've been telling you to put into your tasks, and you can drop these. These are just showing where during your task these events happened. And next thing you can do is you can actually change what's graphed. So by default, we're graphing the right and left hand speeds. But I'll start here. You can see up here, this is all of the data that we collect as part of running the task. So, and these things are going to be about motors and linkages in general. Below the derived channels line, so you can see this little line, it says derived channels. You can go through and see all of these different channels of data that we calculate based on the information that we record. So right hand speed is let's see. So in this case, I've loaded a left arm exam. So the left hand speed is going to be the more interesting thing to look at. But definitely go down, and if you hover over these, you'll see a full description of what it is and what the units are that we're graphing here for you. Something else I will point out. So if I do put the hand speed in, let's see, I'll turn off my events so you can see if I flip this relative signal size button, basically that makes all of the channels display at their full size, which can be useful to help you kind of see more detail on certain things. The other way to show more detail is going to be if you zoom in. So you can just use your mouse, drag, zoom in. So now I've zoomed in, and I can see my hand speeds. See these little blue marks? That's telling you in time where are you zoomed in at in the graph. So I can actually take this, and you can actually see. You can do things like step through. So I can actually step this a kinematic frame at a time and watch things happen if you want. So that's a little bit slow to see, but the line is actually moving. When you're playing with this, you can keep zooming into higher and higher levels of detail if you need. And again, this is going to show you where you're zoomed into. The next thing, let's see, I'm going to zoom back out. Up in the top, this is all of your trials. So with the trials, you're normally in sequential view, so this is kind of as you'd expect. If you press play, it's just going to play through each of the trials as they happened in real life. You can change the view to an overlap mode. Depending on your exams and your data, they might find something interesting here, but you're more likely to find something interesting if you go into a line on event. So a line on event means that whatever the currently selected event is, so this is the target on event, we've taken all of the trials and laid them out such that they all line up on the target on event. So in this case, that lets you see, oh, look, the person's hand speed is spiking at about the same time in most trials after the target goes on. So a couple other little things you can do when you're playing with this data. If you need to, if you wanted to, you can say right-click and say, I would like those all to display red, or I would like those all to display light green, things like that. So you have control to make this look pretty and look the way you would like, which is nice because then I'm going to go back into sequential mode, maybe just pick a small, so here's about 5 seconds worth of the task. So if you want, to make this pretty, and another big use of this is to do things like make a little movie. So here I am going to make a movie of the area I just zoomed in on, so that's my current selection, and I'm going to make the movie of both the hands moving and the graph of kinematic data. So I'll just save that, and then that'll go through and generate a movie that you can take and put in a talk, for instance. So that's a really big use of this tool, is trying to describe, especially to a large group of people that you're talking to that have never seen a Kinarm. It's really difficult to explain your experiment and explain why a certain part of it might be interesting. But if you make a little movie, throw it in your PowerPoint presentation, similar to what we've asked you to do as part of the talks later in the week, then you can really get your point across much more quickly and easily as to what your subjects were doing and why that was interesting. So that's just using the record movie button. There's also a snapshot button. So if you just need a more static picture of what's happening, that's going to allow you to create a static image of whatever is on the screen now. It will just turn that into a PNG, JPEG, or SVG. So SVG being scalable vector graphics. So if you need a vector graphic to put in a paper, then you can get that out of here as well. Beyond that, there's some other tools up here that are worth mentioning. There's a caliper. So if you can just click and drag these calipers around. So if you wanted to do some verification on your task to make sure, were my targets as far apart as I thought they were, or I thought they should be, you can use the little caliper tool and make sure they get, make sure everything measures the way you think it should. Or you can even do some, little vague, a bit of analysis with this and see, okay, how far did the person get perturbed when I used a 5 Newton force, and do some measurements. So if you need them to go further, you can, you can determine that using something like this. As well, there's lots of other visualizations up here that might be useful, like what were the full hand trajectories, or you can turn on and off the subject, add the robot linkages if those are useful to you. And of course you can change the character from a male to a female. We do that automatically based on the selected subject, but yeah. So you have lots of ways you can play. Oh, I should even show that. You can zoom in and out of this. You can, if I turn off that, you can drag it around. So if you want to make a nice picture of something very specific, you can just use the mouse wheel to zoom in and out of a particular area to show something useful, so that when you're making your movies or your snapshots, you can focus in as you would like and as is useful for you to get your points across. So that's a lot about the visualization. There's some other tools that are available in the visualization as well. Like if you want to, you can do a little data cursor in the bottom, and it'll tell you, oh, okay, this speed peak was at a particular time, and it was a particular meters per second. So in this case, 0.49 meters per second at 23 seconds into the exam. So lots of little nice tools like that to, in there for you to make use of. And we'd encourage you to look around and also think about what other tools would be useful for you, because if you find them useful, chances are someone else would. So we always love to hear suggestions. Next thing we'll look at, if you go to this parameters tab, so if you select once you've got your exam selected, you can look in the parameters, and these are all of the parameters that we store about about your exam and about the data. So there's all kinds of stuff in here, like your full TP table and your full all the information about your calibration and your block table, but it's also information on your calibrations and that sort of thing. So this is all information that's going to be available to you in MATLAB. So when you load an exam in MATLAB, you'll be able to see all of this, and Ian will be going over how to load an exam in MATLAB tomorrow. One thing I will point out that can be really super useful when you're doing that is if you right-click on an exam and say copy location to clipboard, then that is going to put the location of that exam file onto the clipboard, which you can then use to help make loading your exam in MATLAB much easier. So now we've gone through looking at an exam. You can also do reporting. So if you've collected Kinarm standard tasks, you can use those. So here I've got a visually guided reaching exam. So I can just say generate a report. And there you go, you've now got a report. This uses all of the exact same code to generate reports as Dexterit-E does. So whether you're generating your report in Dexterit-E or Dexterit-E Explorer, you're going to get the same answers as long as they're matched versions of the two. So with your report, you can save it as a PDF, or you can print it if you want. As well, if this is useful to you, you can right-click on this and say, I would like to extract these images from the report. So if these images are something you would like to throw in a paper, you can say extract images from report, give us a location, and then we'll put those. In this case, I just put them in my documents folder. So if those are something you want to put in a report, that would be how you would get that information out. Next, mentioned a little bit, it's kind of come up a couple times about studies and sessions. So here I've got this data collected as part of a study. So the study was the comparison study, and I've got two sessions. And this is one we did where we collected data on a classic exoskeleton and our newer version of the exoskeleton. So using this study, which is all of these exams were collected as part of this particular session, I can say click on the session, generate a report, and now you can see exactly how someone did in their sensory, motor, and cognitive domains across your this particular session. And if you want to do a longitudinal report, same idea. As long as there's more than one study collected here, hit the report button, and then here is your longitudinal report. This one's a little hard to read because there's a whole lot of data on it, but it's just going to show you between the two exam sessions, how did scores change. You can have as many sessions as you want here. I just happen to only have two, so if you had 5 sessions, it would show you graph all 5 data points for each exam type. So some other useful things to know here. If you've been collecting data, in particular some KST data, some Kinarm standard task data, and you want to get those scores out, make a CSV file for all of someone's scores, you can go to File, Export Analysis. Then you would select an exam type, like say arm position matching. There's lots of filtering options, But here I'm just going to say select everybody. I won't actually do this export because I don't know how long it'll take on my machine. But this would allow you to create a CSV file containing all of the arm position match exams from from your database, and that those CSV files are going to have all of your individual parameter values. So for arm position matching, that's going to be things like your overall accuracy, but then it's going to give you your Z scores and your task scores as well. A few other tools in here that can be handy. This one here, export exams as CSV. You can also do that just by right-clicking and say export exam as CSV. So a lot of people are familiar with MATLAB, but if you are not familiar with MATLAB, if you're much happier working in, say, Python or Java or R or something like that, this allows you to export all of the kinematics, all of the parameters, everything we have into a CSV file. We have a couple of examples on our website about how to read one of these CSV files in Java or Python, but it's a reasonably formatted CSV that just kind of contains huge columns that contain all of the kinematics that you would need to analyze. So if you want to use a different language, export your exams either one at a time or as a group to CSV, and then you can process them in whatever language you would like.